Hi, I'm Willie Williams, one of 3 members of Group 4. I'll be followed by Jesse Campbell and Joey Lamond, and we're going to be presenting emotion today. Emotions. We all have them. While emotional cues such as happiness, anger, fear, or sadness are commonly perceived on face value across all cultures, the anatomical structures and physiological changes associated with emotion are just now being understood. Now, beginning in 1862, French neurologist and physiologist G.B. Duchenne de Bellone was the first to systematically examine the contributions of cranial muscles to facial expression. Now see, he was curious to understand how coordinated contractions of muscle groups in the face express pan-cultural emotional states. To accomplish his goal, he used a process known as spheroidization, or electrical stimulation, to activate single muscles and groups of muscles in the face, dorsal surface of the head, and neck, and would document his findings via photography. It was his contribution which was key in identifying the muscle groups such as the orbicularis and oculi that are not easily controlled by force, but are, quote, put into play by the sweet emotions of the soul, as he put it. The natural smile will be termed the Duchenne smile. Duchenne concluded that emotion-driven contractions of muscle groups surrounding the eyes, along with the zygomatics majors, conveys genuine expressions of happiness and joy and laughter. It was also observed in his experiment that there were key differences in muscle contraction between a forced smile produced by willful contraction or electrical stimulation and a spontaneous emotional Duchenne smile. Fake smiles, also known as pyramidal smiles, were driven by descending pathways of the motor cortex, which communicates with the brainstem and spinal cord by pyramidal tracts, while the real smiles, also known as Duchenne smiles, were motivated by motor areas in the anterior cingulate gyrus that accesses the motor circuitry via the extrapyramidal tracts through the brainstem reticular formation. Now, patients with damage to the descending pathways of the motor cortex further validated Duchenne's study. Now, when asked to smile on command or give a fake pyramidal smile, these patients could not. These patients had difficulty moving their lower face muscles on either side of their face. However, given the opportunity to express a genuine smile, these patients had no difficulty whatsoever, as other regions of the forebrain remained intact. This syndrome would be termed voluntary facial paresis. On the contrary, emotional facial paresis was the exact opposite. Whereas the motor cortex has no injury, one can express forced fake smiles and facial expression, but a genuine smile or facial expression could not be noted because their descending pathways in the non-classical motor cortical areas were disturbed. Now, while most people associate emotion with outward facial expression and behavior, it is much more complex than this. Now, before it was believed that emotional responses were grouped solely under the limbic system. It is now known that in addition to the limbic system, which includes the amygdala and cortical areas in the orbital and medial aspects of the frontal lobe, that regions in the forebrain and diencephalon motivate lower neuronal pools associated with somatic expression of emotion as well. Together, these structures work in unison to make up the complexity of the emotional motor system, which is fundamentally key in regulating goal-directed behaviors, rational decision-making, moral judgments, and interpreting other people's social behavior. Now, as it pertains to the physiological changes, the most obvious signs of emotional arousal involve changes governed by the autonomic motor system, which is further divided into the parasympathetic and sympathetic motor systems. Thus, increases in heart rate, sweating, blushing, or turning pale, hair standing on end, also known as piloerection, or the certain urge to go to the bathroom, all can be attributed to changes in the sympathetic and parasympathetic and enteric components of the visceral motor system, which moderate smooth muscle, cardiac muscle, and glands in the body. Now, Walter Cannon, an American physiologist, once argued that increasing the activity of the sympathetic division prepares an individual to fully use their metabolic reserves, or energy, and other resources in challenging situations. Now, on the flip side, activation of the parasympathetic and enteric divisions promotes the buildup of those metabolic reserves. This led to the belief that activating the sympathetic nervous system was an all-or-nothing process, where given an intense encounter, all resources would be expensed from within the sympathetic division. It is now believed that due to recent research that physiological responses to emotion can be quite multifaceted and don't just involve one division of the autonomic nervous system per encounter, and are mediated by the forebrain. For example, the need to go to the bathroom, parasympathetic, when terrified, sympathetic, of a loose grizzly bear and your heart's racing, which is also sympathetic. Now, what happens in these situations? Well, we now know that the neural activity of these complex situations is relayed from the forebrain first by the hypothalamus to the brainstem reticular formation, then to the visceral and somatic nuclei, thus influencing the autonomic nervous system. So, how do we know that facial expression associated with emotion is tied to the autonomic nervous system? Well, recent studies have shown that when individuals naturally express facial patterns of anger, disgust, fear, and happiness, highly specific differences of heart rate, skin conductance, and temperature can also be measured and reproduced within the visceral motor system. It is believed through these findings that when the natural facial expression associated with emotion is produced, signals in the brain not only engage the motor cortex, but those circuits produce emotional states as well. This means that one source of emotion is sensory influence from our muscles and internal organs, and that emotion and sensory motor behavior are intrinsically linked. In 1928, Philip Bard set out on a course of experiments that would prove that the hypothalamus was the center for the coordination of visceral and somatic motor components of emotional behavior. He anesthetized and removed both cerebral hemispheres in multiple cats. When the anesthesia had worn off, many of the cats behaved enraged, and some remained docile. The enraged cat's behavior was termed sham rage because there was no obvious target of the anger. The autonomic characteristics of anger, such as increased blood pressure, heart rate, dilation of pupils, and piloerection were noted as well. However, Bard had found that sham rage only occurred if the causal hypothalamus was still intact. Sham rage could not be noted if the hypothalamus was completely removed. He found that while the subjective experience of motion would depend on an intact cerebral cortex, the expression does not depend on the cerebral cortex at all. In addition, due to its placement in the brain, emotion was phylogenetically an older part of the nervous system. Basically, this meant that the body's natural reaction to life-threatening situations was to enact visceral and motor components to ensure survival, which on the surface, at face value, displayed some type of emotional behavior. Additionally, another experimenter, Walter Hess, created studies supporting Bard's findings with the use of electrical stimulation and various portions of the hypothalamus of cats as well. Except for these cats, they were awake. Now, some portions were stimulated, causing defensive postures and even fear. Both experimenters' work led to the conclusion that the organized behavior accompanied by emotion are in the diencephalon and the brain structures connected to it. Now, the way in which the hypothalamus influences the visceral and somatic motor system are highly complex. The main target of the hypothalamus is the reticular formation. The reticular formation contains over 100 identifiable cell groups and receives information from the hypothalamus. It then provides output to both somatic and visceral motor effector systems in the brainstem and spinal cord to control cardiovascular function, respiration, urination, vomiting, and swallowing. The neurons in the reticular formation produce widespread visceral motor and somatic motor responses and have the ability to override reflex function and also have the ability to involve every organ in the body. Additionally, there are descending projections from the forebrain to the reticular formation that contribute to emotion and the expression of somatic and visceral motor behavior as well. These descending projections, which control emotional expression, are considered a part of the limbic system, but are located outside of the classical motor cortical areas and entail two parallel systems that are functionally and anatomically distinct. The voluntary or volitional motor component is made up of classical motor areas of the posterior frontal lobe and related circuitry in the basal ganglia and cerebellum. The descending pyramidal and extrapyramidal projections from the motor cortex and brainstem are responsible for the impulses associated with voluntary movement and terminate on the reticular formation and motor neuron pools, while the descending pyramidal and extrapyramidal projections from the limbic system run parallel and are in the ventral medial forebrain and hypothalamus and control emotion. Both projections work in unison with the brainstem and reticular formation limbic system to project the output of emotional expression.
Good afternoon. My name is Jesse Campbell. I am going to talk to you about the limbic system and its connection to emotional response. The visceral motor system is meticulously connected to emotional response. Because of this, emotional expression necessitates central brain structure, action that controls brainstem and spinal cord autonomic preganglionic neurons. The sophisticated functioning neural cores that manage expression of emotion are categorized as the limbic system. Let's take a look at the historical development of the limbic system. In the late 1800s, Paul Broca labeled the part of the cerebral cortex that makes a medial loop around the corpus callosum and diencephalon, the limbic lobe. At the time, he considered the sense of smell to be the main sensory input to the limbic lobe. Although Broca's hypothesis was not complete, smell is one of the important inputs to the limbic system. In the early 1900s, James Papez introduced there are explicit circuits in the loop of the limbic system for understanding and response to emotion. This was called the Papez circuit. The cingulate gyrus and hypothalamus connected by the mammillary bodies to the anterior thalamic nucleus. The cingulate gyrus also connected to the hippocampus, and the hippocampus connected through the fornix to the hypothalamus, completing the loop. Research has shown that this was an incomplete representation of the actual circuit. The complete circuit also includes the orbital and medial prefrontal cortex, the ventral portions of the basal ganglia, the mediodorsal nucleus of the thalamus, not the anterior nucleus of the thalamus, like Papez proposed, and the amygdala. Although the parahippocampal gyrus is connected in the circus, the hippocampus itself has been found to have little to do with emotional behavior. There have been experiments to show how the limbic system is the effector and coordinator for emotions. Let's take a look at some. Heinrich Kluver and Paul Bucy conducted experiments destroying the limbic system by removing it from rhesus monkeys. The experiment was initially set up to study the effects of mescaline on the overall actions of the brain. Mescaline caused the monkeys to lick and chew their lips, which Kluver and Bucy knew to be like temporal lobe epilepsy. They took out the temporal lobes of the monkeys and noticed no fear or aggressive behavior, leading to follow-on experiments replicating the same results in even the most aggressive monkeys. This showed the effects of the limbic system holistically. The most important realization of the limbic system post-Papez was the amygdala. John Downer, he removed one amygdala from rhesus monkeys and disconnected the optic chiasm and commissures that link the brain hemispheres. He then covered one of the eyes. And this created monkeys that had only visual information from one side. Some monkeys had the information from the amygdala on the same side and others on the opposite. Those that had vision on the same side as the missing amygdala had a subdued response, exhibiting no fear and no regression. The monkeys that had vision on the opposite side, vision on the side with an existing amygdala, exhibited fear and aggression, just like a completely intact monkey would. Also, this was only limited to visual input. If either monkey received touch input, aggression and fear responses were intact. This showed more details of the limbic system, specifically the amygdala. The amygdala, anatomically, is the most inferior portion of the limbic system and is located anterior to the hippocampus. The amygdala is split up into three main groups: the medial group, the central group, and the basolateral group. The medial group is connected to the olfactory bulb. The basolateral group is connected to the orbital and medial prefrontal cortex of the frontal lobe and the associational cortex of the anterior temporal lobe. The central group is connected to the hypothalamus and brainstem. The amygdala receives cortical inputs, defined as highly processed inputs, as opposed to the hypothalamus, which receives unprocessed inputs. The basolateral group of the amygdala is connected in a triangular circuit between the medial dorsal nucleus of the thalamus and the orbital and medial prefrontal cortex. This allows interactions between the amygdala to the prefrontal cortex and to the thalamus. For the thalamus, it is through the basal ganglia. The amygdala is not only a location that facilitates fear and aggression, but it is also a location that these responses can be learned and developed. The amygdala can participate in creating associations between different sensory stimuli. The associations, or this allows for the associative learning process to take place in the amygdala and allows for stronger connections based on postsynaptic neurons being activated when inputs are received. The amygdala will signal integrative centers that elicit relevant behavior of reward seeking and punishment avoidance. Joseph Ledoux and colleagues tested different parts of amygdalas in rats. The tests were focused on conditioned fear responses. The test delivered a shock after a sound, which conditioned the rats to elicit a raise in blood pressure as well as a freezing, non-movement fear-evoked response. They first found in the brain that removal of the entire medial geniculate nucleus deleted any fear responses. But when the connections between the medial geniculate nucleus and the auditory cortex were removed, there was still a fear response. The only part left to test of the medial geniculate nucleus was the connection to the basolateral group of the amygdala. When this connection was removed, there were no fear responses. They also found that connections from the central group of the amygdala to the midbrain reticular were necessary in fear responses. A specific disorder seen in humans, Urbach-Wiethe disease, has many symptoms, one of which is damage to the amygdalas. In the 1990s, there was a patient, initials SM, studied that elicited no fear responses when confronted with fear-evoking situations such as encountering snakes, spiders, and well-crafted haunted houses. Also, she was unable to identify fear in images of faces that showed physical response to fear, but able to identify all other emotions in faces. In fact, SM was unaffected emotionally by these situations other than a, quote, piqued curiosity and aroused excitement in a manner similar to the feeling she gets when riding a roller coaster. We've seen the detrimental survival and social effects of losing the amygdala, but there are also deleterious effects with overstimulation of the amygdala. There has been research in the dichotomy between monozygotic and dizygotic twins showing that there are heritable neurobiological origins for unipolar depression. This is when there are abnormal patterns of blood flow that are apparent in the triangular circuit interconnecting the amygdala, the medial dorsal nucleus of the thalamus, and the orbital and medial prefrontal cortex. The more popular treatment for unipolar depression is selective serotonin reuptake inhibitors. The limbic system is a loop system of nerves connected in the brain that control the emotional translation of experiences and responses. The amygdala is critical to processing emotions, specifically fear and aggression, the keys to survival in any species, to include humans. Thank you. This concludes this portion of the presentation on emotional expression. Have a good day.
Good afternoon, class. As you know, my name is Joseph Lamont, and today we will be talking about emotions. In particular, we will be discussing the relationship between the neocortex and amygdala, cortical lateralization of emotional functions, emotion, reason, and social behavior, as well as emotional reinforcement and addiction. So we will begin our discussion today in regards to the relationship between neocortex and the amygdala. We have to understand the pathways are not completely understood, but one thing is for certain: the amygdala, neocortical areas, and subcortical structures are critical in emotional processing. So what does this mean? In order to answer that question, we first have to go over anatomical structures. And as you see on the top right-hand corner, we have the amygdala here, the neocortex, the prefrontal cortex, the nucleus accumbens, which we will talk about later in the discussion, the ventral tegmental area, which we will talk about later in this discussion as well as the hypothalamus. So it's connected to the hypothalamus, brainstem centers, orbital and medial aspects of the frontal lobe, so prefrontal cortical fields, thalamus, ventral portions of the basal ganglia. So it's important to note that the amygdala emerges as a key node in the network that links together the cortical and subcortical brain regions involved in emotional processing. So now that we have basic understanding of the anatomical structures involved with the relationship between the neocortex and amygdala, we are going to discuss one of its jobs. And one of its jobs is decision making. It's essentially a shot caller, meaning the amygdala is connected to the prefrontal cortex and basal ganglia, and it influences the initiation of reward-seeking behavior or punishment avoidance behavior. And because of this, it may lead to behavior modification. So if I do something and there is a reward or benefit of doing it, I will most likely continue doing it. And vice versa, if I do something and it's punished, I will avoid doing that thing. So now the question arises, how does the amygdala influence future behavior? Well, the prefrontal cortex and amygdala organize and plan future behaviors by providing emotional input to covert or overt these deliberations. In continuation of these emotions, we ask ourselves the question, how do feelings arise? Feelings are actually a consequence of self-awareness, meaning the combination of implicit processing and explicit processing. Implicit processing is going to be immediate. It's your experience in that moment. If I were to be punched in the face, the immediate reaction is going to be, okay, I've just been punched in the face, and the pain receptors, I feel the pain, feel everything that's going on around me. And then the explicit, the semantic, well, what does this mean? Oh, well, this person's angry at me. They're angry at me, they have hurt me, and now I am angry at them, and I will react accordingly. So that's the implicit and explicit response. So finally, we have the amygdala and the neocortex, which make up subjective feelings that attend most emotional states. Another thing that I found very interesting is that we have emotional working memory. As mentioned in the previous slide, we have a triggering experience, the punch in the face. And if you look at the top right corner, that stimuli is going to be processed in two ways, the implicit and explicit, right? So we have the amygdala dependent associative learning, and then the hippocampal dependent explicit memory. So the working memory works by sustaining neural activity related to processing elements of emotion experience. But that makes sense, right? If we didn't have that, we would have the emotional range of a goldfish. We would forget what we were feeling every 5 seconds. Our next topic of discussion is cortical lateralization and emotional functions. Now, what is cortical lateralization? It is the specialization of one side of the brain or the other. As most of us already know, the right side of our brain will control certain functions and process certain stimulus, and the left, and they could be similar or very different. If you look at the top right corner of the screen, the left side of the brain, left hemisphere, has a speech center. The right side has an auditory cortex for the left ear, and the right side has an auditory cortex for the right ear. So we talk about left hemisphere versus right. As previously mentioned, the brain is asymmetrical in function. It doesn't have the same functions on both sides, and this holds true for even emotion. And that could mean happy movements by how our face moves, sad thoughts, is one side more associated with positive or negative emotions, speech, how we elicit emotion in speech, and we'll discuss all those things in further slides. Continuing our discussion on right versus left hemispheres, it is important to note that the right hemisphere is vital for expression and comprehension of speech. And if you look at the top right corner of the screen, we have the suprasylvian sulcus and gyrus, and the anterior portion of the parietal and the posterior portion of the frontal lobes. And so understanding this anatomy is important because if we have a patient that presents to us, a blow to the head, get stabbed, whatever type of trauma or a neurological disorder where they develop a lesion in this area, they lose their ability to modulate speech patterns. They become monotonous. They will talk like this, and if they would like to express an emotion, they won't be able to do so with their speech. They would actually have to tell you, I'm very upset by this, or I'm very happy with this. And so this condition is called aprosodia. So this information leads us to what I think is kind of the fun part. We have discovered that the left hemisphere versus right hemispheres are more associated with a certain type of emotion over the other. The left hemisphere is more associated with positive emotions, and the right with negative. So if we feel joy, love, pride, hope, those are positive emotions. Those are going to be associated processed in the left hemisphere. And jealousy, doubt, anger, envy, sadness are going to be associated on the right. And interestingly enough, if we have a patient that has a lesion on the right side of the brain, they are more likely to be unduly cheerful or inappropriately optimistic. So they may not feel as sad at a funeral, or vice versa. People who have lesions in the left side of the left hemisphere are significantly more likely to suffer from depression. Now that we have discussed emotional processing as it pertains to positive and negative emotions and what side, which hemisphere those are processed on, and expression through speech modulation, we're going to discuss how individuals are more readily fully expressing emotions with a certain side of their face over the other. And so if you look at these pictures of these famous people, or a person who was painted and became famous because of the painting, we see that individuals are more readily expressing their emotion on their left side before their right. And this is because the right hemisphere is superior at this. The right hemisphere is better at picking up emotional nuances in speech, and same thing with facial expressions. It is more readily going to perceive nuances in facial expressions as well. So now we are going to move on from processing and expression, and how do we apply it? The next topic is emotion, reason, and social behavior. So the amygdala and the prefrontal cortex, along with their striatal and thalamic connections, process emotions, but also perform an integral task to social behavior, which is rational thinking. So what does this mean? Well, rational thinking is taking those emotions that are processed, or the stimulus we pick up from other people, or the social cues, whether it be facial, speech, body language, etc., and we appraise the circumstances and we apply it. So if I see someone who's sad, I don't go up to them, or I shouldn't anyways, go up to them and make them sadder. I'm going to try to console them. If I see someone happy, I want to add to their happiness. It's because I have processed those emotions, and now I'm going to implement it in a social setting. Our next topic of discussion is emotional reinforcement and addiction. Before we get into how addiction is chemical, we first must understand some anatomical structures. I would like to point out the caudate nucleus, the putamen, the nucleus accumbens, and the ventral tegmental area. The ones we will be focusing on right now are the nucleus accumbens, NA for short, and the ventral tegmental area, VTA. And this is the primary site of drug interaction for emotional reinforcement. So now we ask ourselves, what is the process in which drugs reinforce emotions and create addictions? The top right, you'll see a figure where we have a dopamine release, different drugs like cocaine, cannabinoids, opiates, alcohol, and it's going to do this in two ways. It's going to prolong the action of dopamine in the nucleus accumbens, and potentiate the activation of neurons in the ventral tegmental area and the nucleus accumbens. I appreciate your time. Hope you enjoyed our video.